Hi, I'm going to get started. Good afternoon to those of you joining us at the Centers of Excellence in Hematology Metabolomics Mondays, um, Multiomics Mondays, I guess I should say. Today we are privileged to hear from Alan Maschek. How do I say your last name? Maschek?
Yeah, Maschek. Even
Maschek,
people in
thank
my
you.
family disagree. Yeah.
That's okay. I like that. Anyway, Alan comes to us from the University of Utah Metabolomics Core, where he is the director of the lipidomic analysis that's being performed there. And today he's going to teach us all things lipidomics. And he has agreed to have questions come up during the talk. We are recording this and it will be posted on the CCEH's YouTube channel if you want to watch it again. And so we should go ahead and begin, Alan, and feel free to interrupt with questions, or you can write your questions in the chat and I'm happy to ask them for you or invite you to ask them. And thanks for your attendance.
All right. Hi, everyone. Happy to be here and give this presentation for you all now and all the people in the future who are watching. I'm just getting over a cold, so if my voice starts to waver later on here, bear with me. I do have a nice cup of tea to for my throat. All right, so I'm gonna touch on some real big picture lipidomics ideas and what we do here and how to interact with us and break into some of our different platforms that we offer, what you can expect from reports and informatics, and of course, cost and sample submission, and maybe most important, how uh, researchers can prepare their samples to get ready for lipidomics. I created this contact info slide for posterity, so you can screen grab that. And yeah. All right. So I'm the associate director of the Metabolomics Core. I've been here for over 11 years. My initial background was in natural products discovery, so which is kind of a mix of organic and analytical chemistry, and transitioned to the core after a postdoc here in the School of Pharmacy. So wanted to, at the top here, is to highlight a few key things, some take-home points, how we run lipidomics. We support both untargeted and targeted LC-MS workflows. On the targeted side, our method includes well over 2,000 transitions with potentially 10x more in theory. But in a real run, we typically report anywhere from 600 to 900 unique lipids per run. We've built additional targeted panels that cover nearly every major lipid class. Some of these require class-specific approaches, and I'll touch on that a bit later. Our data are quantitative, and we rely on a technique called isotopic dilution, where we spike in labeled internal standards that are tailored to each class, and this helps us normalize across a very diverse lipidome. We're very flexible on sample types. We work with just about anything. Once the data has been acquired, we also provide bioinformatics support with process outputs along with PCA, volcano plots, and lipid class enrichment to help interpret what you're seeing. And to run all this, we have 4 LC-MS systems split between Agilent and SciEx vendor platforms, which gives us a lot of flexibility in how we approach each project. Emerging in the early 2000s, lipidomics is a branch of metabolomics that focuses on the comprehensive analysis of lipids within a biological system. As essential components of cell membranes, energy storage, and signaling pathways, lipids play critical roles in physiology and disease. Unlike other biomolecules, lipids exhibit remarkable structural diversity, making their identification and quantification complex. Advances in mass spec and bioinformatics have driven lipidomics forward, enabling high throughput profiling of lipid species across various biological contexts. And then by integrating lipidomics with other omics, like that's what we're here to do, researchers can uncover lipid dysregulation in diseases such as cancer, neurodegeneration, and metabolomic disorders. Okay, so step one, I know James touched on this. Really, the first step is to have a conversation, to reach out by email to discuss your study goals, sample handling, timelines, cost, and overall expectations. That initial communication is key. All right, so let's get into the lipidomics processes. First, let's talk about how researchers the best handle their samples before they even get to the core. So when it comes to lipidomics, how you handle your samples before analysis can make or break your results. Lipids are highly sensitive to degradation and oxidation, so proper collection, storage, and processing are critical. First and foremost, consistency is key. Always collect samples under the same conditions to minimize variability. Use glass or low binding plastic tubes. In practice, we use low binding plastic tubes. Glass is great, but it's expensive and kind of difficult to work with. And whenever possible, snap freeze your samples in liquid nitrogen to prevent enzymatic activity. Store in -80 and avoid repeated freeze-thaw cycles. For plasma or serum, the fasting samples are ideal to reduce dietary lipid interference. Plasma is the preferred matrix over serum, but serum works just fine. And if you're working with tissues, standardizing dissection and the handling times is crucial. Replicates. The number of replicates needed in a study depends on the sample type and experimental design. Lipidomics data tend to be more variable than other omics data due to sensitivity to handling and the temperature and the buffers, so consistency is critical. For well-controlled samples, we recommend a minimum of 5 biological replicates per group. If for animal studies, if they follow similar guidelines, like if variables like age and diet are controlled, 5 also works. For biofluids or tissues, the minimum is 5. We'd like to get up to 10. And more for heterogeneous tissues. In human studies, due to the high individual variability, you plan for 20 to 30 replicates at a minimum. And then finally, a small pilot project, a pilot study can help determine the appropriate sample size on your specific system. And maybe more importantly, they also help dial in the internal standards and inform what lipid classes are observed and how we can tighten up the experiment in general. How much material do you need? Well, thanks to the sensitivity of modern LC-MS methods, only a very small amount of material is needed, but it needs to be the right material. 2 things to keep in mind. First, even a small sample must accurately represent the whole. And second, you only need enough for the planned analysis. Too much material can reduce reproducibility and affect the extraction efficiency. Tissue masses, cell counts, or protein quantification are essential for normalization, and samples must be precisely measured, flash frozen, and transferred or shipped on dry ice. At the core, we can run a BCA for protein quantitation, but cell counts and accurate masses are what we prefer. Also, keep the vial and sample name labeling simple, ideally 3 to 4 digits, to streamline the tracking and avoiding any transposition errors. And if you want to run metabolomics and lipidomics, then yes, you can send one sample, and we would split that here at the core at the time of sample processing. Okay. This is kind of a very simple overview of just how we extract our samples. What we want to do is basically isolate the lipids from protein, salts, and other extracellular material that can interfere with the analysis. This is usually done with a liquid-liquid extraction and partitioning an organic solvent like chloroform, methanol, or MTBE that contain internal standards against water or PBS. This solubilizes and separates lipids into a clean organic phase for consistent, reliable measurement. We also employ solid phase extraction, or SPE, for high throughput sample preparation, or when some lipid classes need to be enriched, like oxylipins. Okay, so let's briefly touch on instrumentation. LC-MS is a hyphenated workflow. Liquid chromatography separates the lipids, and then the mass spectrometer, or MS, detects and quantifies them. Samples are introduced via the LC stack here, carried by a mobile phase, and fed directly into the mass spectrometer. So let's now breaking down the mass spec platforms here. An untargeted lipidomics workflow on a QTOF, stands for quadrupole time of flight, focuses broadly a broad profile of all detectable lipids in a sample without prior selection of specific targets. QTOFs provide high resolution, accurate mass data, allowing for comprehensive lipid identification and discovery. This workflow is ideal for hypothesis generating studies, biomarker discovery, or examining global lipidomic changes across conditions. In contrast, a targeted lipidomics project on a triple quad is designed to precisely quantify a predefined set of lipid species with high sensitivity, high dynamic range, and reproducibility using multiple reaction monitoring, or sometimes called selective reaction monitoring. The triple quads selectively detect known lipids based on their mass transitions that optimize for sensitivity and throughput. This approach is best for validating findings, studying specific metabolic pathways, or performing absolute quantitation with internal standards. So while the QTOF excels at discovery and broad profiling, the triple quad is the gold standard for precise, high confidence quantitation of known lipids. Okay. The LC side for lipids uses a couple of different approaches depending on the lipid class and analysis goals. Reverse phase, or sometimes you'll see it called C18 or C8, separates lipids based on hydrophobicity, making it ideal for complex mixtures of neutral and nonpolar lipids like triglycerides and phospholipids. Helic or hydrophobic interaction chromatography separates based on head group polarity, making it well suited for distinguishing phospholipids and other polar lipids. And then in contrast, shotgun lipidomics skips chromatography entirely and directly infuses the extracts into the mass spec. This allows for high throughput profiling, although it suffers from ion suppression and limited separation, making it less reliable for complex or isobaric lipid species. So you'll see these reported all in the literature. They all work and have their pros and cons. We tend to rely on reverse phase chromatography, and we supplement that with some Helic chromatography. Okay, so in reverse phase chromatography, and this is a chromatogram here where we have time and intensity, time on the x-axis, intensity on the y-axis. Lipids elute based on hydrophobicity. So the first compounds that come off the column are more polar, water-loving metabolites and lipids, followed increasingly by non-polar lipids. So we'll get acyl carnitines, lysophospholipids, and fatty acids, fatty acyls eluting early on, followed by phospholipids, PS, PI, PG, PE. Mixed in there is then sphingolipids, gangliosides, ceramides, sphingomyelins, and then followed by diglycerides, cholesterol esters, cardiolipins, and triglycerides. So the broad lipidomic screens are great for covering major lipid classes, but they often miss or suffer for looking at specialized types like acyl carnitines and oxylipins. That's because these lipids differ in polarity, abundance, and stability. Acyl carnitines are small and polar, while oxylipins are reactive and degrade easily. So standard methods do not retain or ionize them well. So we use dedicated LC-MS methods with tailored chromatography, ionization, and sample handling to get accurate data on these tricky but very important lipid species. Here's a summary of our class-specific targeted lipidomics platforms that we've created. Sphingolipids and acyl carnitines can be analyzed using the standard extraction, meaning that we can run both broad coverage lipidomics and sphingolipids and acyl carnitines from the same sample prep, so we don't need additional samples here, and we do not charge extra for their runtime. In contrast, though, oxylipins and bile acids require a dedicated sample preparation due to their distinct chemical properties. So to summarize up to this point, our standard lipidomics workflow provides broad coverage. It works well for larger acylcarnitines, phospholipids, major fatty acids, sphingolipids, glycerol-lipids. Oxylipins and bile acids are offered as an additional service and may require extra sample material. We work with just about any biological material, and as a rule of thumb, if you can see it, then it's probably enough material. We recommend a minimum of 5 biological replicates in general, and we also offer a high throughput plasma lipidomics assay covering over 600 lipid species. I won't go to that and today into any detail, but happy to chat about that anytime.
Alan, will you go back one slide? This is
Sure.
Diane. I don't know what PUFAs are.
Polyunsaturated fatty acids.
Thank you.
Yes.
Okay.
And that's probably a bigger class, right, than what's shown here. Yeah, PUFAs. Okay. And let me transition now to talk a little bit about our data and our reports and what you can expect. We provide summary, background, methods, and other experimental details, data tables, and a basic statistical analysis. Our turnaround time is very seasonal and project dependent, 4 weeks up to 6 months. Okay. Lipidomics reports and bioinformatics. So lipidomics data analysis differs from metabolomics in that lipids behave in coordinated patterns, not necessarily and not usually as isolated molecules. So entire lipid classes often shift together, reflecting a shared biosynthetic pathway. For example, changes in phosphatidylcholines often mirror those in phosphatidylethanolamines and phosphatidylserines. If we're looking at isolated PCs, for example, we may not reach a significance for those individual molecules, but taking as a whole, the whole group may have a significant p-value. And at the same time, acyl chains may be remodeled across classes, meaning a shift in fatty acid composition in one class often shows up in another. So this creates a layered data set where both class-level trends and cross-class acyl chain dynamics matter, making lipidomics analysis more about understanding networks than individual compounds. We like to use volcano plots because they show both statistical significance here in the y-axis and directionality via fold change. So things here on the right are higher in the experimental group, lower in the experimental group on the left, or conversely higher in control. And we also like to color code by lipid class, highlighting class-specific trends, making it clearer for a compelling visualization. So all the lipid classes are shown here, and it's a little bit hard to see when we have 10 lipid classes, but we can isolate those and only focus on phosphoinositols, for example, and toggle through these and highlight different ones and see some nice trends that way. This custom R script was created by our bioinformatic team led by Paul Stewart, who helped put this together. My second favorite analysis is an enrichment analysis. So earlier, I just mentioned the importance of interpreting lipidomics data across multiple layers, not just individual species. So we can look at broader patterns like class level shifts and acyl chain features. This is where enrichment analysis becomes valuable. So here, this is an output of a tool called LION, which stands for Lipid Ontology, and where it maps individual lipids into biological meaningful categories or terms that are based on structure, function, subcellular localization, and lipid class. And it runs statistics on hundreds of these terms, and then will generate what features are significantly up and what features are significantly down. I know you won't be able to see these terms there. And so this list will help you move from a flat lipid data table into a more deep interpretable insight about the metabolism. And we've also created bubble plots of this enrichment data from Paul's team that we kind of like that look a little nicer here. Okay. And then now using the cores, we have an online sample submission. Of course, we want to first have a conversation at the core website. It starts with getting a work authorization. If you've never used the core before, we need to first set up an account, and that'll lead you through submitting those samples. And then finally, costs. The project costs are dependent on where you fit into the university tiers. CIHD members have their own rates, as do U of U researchers and external academic researchers. I have an example here for using CIHD external, which I think many from this group might qualify for. So in this example study, where there was an N of 48, the approximate cost would be about $4,800. So that breaks down to a sample cost of $71.52. That includes all the internal standards, all the consumables, all the instrument time, all the buffers that go into that. And then on top of the sample cost is the data analysis, which would be pretty close to 8 hours for a project of this size. And that's kind of what you can expect with that cost. Like I mentioned, if this is for broad lipidomics, if we wanted to add oxylipins, for example, that would be an additional $3,400. Some acknowledgments and references. Michael Holkopek and Kim Ekrus came off with this really great mass spec book for lipidomics. They're two great websites for lipid data and lipid informatics, lipid maps here, and this lifesciencetools.org are two of the great resources. And I'd like to give a shout out to our course admin team, Paul Stewart, our bioinformatics specialist, and my lab specialist, Trevor Lonergan. So that was a pretty quick presentation there. So I'm happy to answer any questions.
So, Alan,
Yeah.
have you done samples for people on hematopoietic stem cells or anything that's in the blood lineages?
Sure, we have. We have. I I mean, big
I'm just
picture, they
wondering, I guess my question is, um, you're still on that pretty 100,000 cells or something like that. Is that what your thing said? Trying
yeah,
to,
and that's a very you know, cells, of course, are extremely diverse from the type. So there, 100,000 cells is a great starting point to get a very nice broad coverage. For some major lipids, we could probably go well below that, right? Because the dynamic range of the lipidome is quite large. So for getting at the top 100 lipids, I think we could probably get it with 10,000 cells. But to get a deep dive of those, we want more.
I'm just thinking about it in the context of if somebody's got an animal model,
Yeah.
are you looking at small populations of cells? Exactly. Can they do this? I guess is the first question.
Mhm.
And I don't know the literature on that, or if anybody else wants to chime in and ask their questions regarding it. I was trying to sort of think of questions that might come about for, the centers of excellence in non-malignant hematology, what they might be looking at. I know I've done lipidomics with you, but that was on liver samples. And so I had more sample
Yeah.
than blood samples. Does anybody have questions? You're welcome to unmute yourselves and ask away. You have the captured attention of Alan right now, or you can think about it and then reach out to him again later if you have more questions. Anybody? This is actually kind of a quick summary. I think we've got anybody chiming in. Nobody's raising a hand. Okay. Well, thank you all for your attention, and thanks, Alan, for the
Thank you.
presentation.
My pleasure.
And we'll post this presentation on the CCEH's YouTube channel so that you can watch it again if you have questions. And maybe the most important slide could have been the first or second slide where it has the contact information for Alan, so that if you have questions or have a desire to pursue lipidomics in your experimental models, the Utah Center is ready to help. And that's it. So thanks, everybody, and take care.
Okay, thank you.
Thank you.
We'll be in touch.
Okay.